So the immune system has this incredible ability to recognize something absolutely specifically at a molecular level. Now, the process by which this occurs comes back to the cell that we call the dendritic cell, that one that has all those little hairy things on it. Here it sort of looks, I guess, like a Darth Vader or something, as a sort of primeval cell. But what it does is it actually here, all these little blue things are dendritic cells in the skin. In fact, dendritic cells form 25% of the cells in the skin, just to show you how important the immune system is. And even though we don't think of the immune system as an organ, let's say a bone or a spleen, it functions as a system throughout the body. And in fact, these cells actually inhabit part of the lymph gland over here rather than the whole one. So they're localized in certain areas, they spread out in certain areas, but they take up a microbe or a bacterium or part of our own tissue, chew it up, and I'll show you how it puts it on a molecule that shows it to the immune system to instruct the immune system in this recognition process. And in fact, there are two key molecules in this recognition process. The MHC is what we call the HLA antigen, or it's a major histocompatibility. It doesn't matter about the words. It's a molecule that's on the surface of a dendritic cell. And in the case of a T cell, there's part of the T cell called the receptor. What happens is that the foreign peptide, which is part of a protein, is processed by the dendritic cell down here, put in these little clefts over here so that it has a molecular fit. And then the T cell receptor sees another part of that molecule, and just imagine every imaginable shape you can think of, and there's a cell in the body that can recognize all of these different shapes. In fact, there are billions upon billions of different cells in the immune system, each of which can recognize things down to a little eeny-weeny bit of a molecule. So this is how what we call immune recognition occurs. And these cells then will begin to act when they recognize what it is they're instructed to recognize. And we'll talk about the molecular basis of how that happens. But this is, again, a stick diagram, and we like drawing these. This is the T cell receptor. This is what we call major histocompatibility complex. And this is the peptide, or a little bit of a protein, that's right in the middle. And you can see that it's sort of like a baseball glove that fits it in, and the T cell then recognizes part of it. That's looking sort of end on into the molecule. That's looking across, and you can see in yellow the peptide. And in fact, if you look kind of down on to the MHC, this here is part of a protein. We call that a peptide. In the case of what we call class 1, which is what the killer T cells recognize, they recognize just a little bit, about 7 or 8 of those amino acids, which are the building blocks of peptides. Whereas class 2, which may be more important in the case of MS, can recognize a whole string of these, maybe as long as 20 to 30 amino acids. So this is actually looking down on that molecule when it's actually bound the peptide over here. This here, for example, is a model of that peptide, and they've sort of added on in blue here the little bits of the other molecule that help them lock together in that recognition process. So what happens once the dendritic cell picks up that bit, chops it up, and presents it to the T cell? Well, it may instruct certain cells called CD8 positive T cells. 8 is just a molecule on the surface. And those cells, once they're instructed to recognize that little bit of protein, if they see it again on a cell, will then go ahead and kill that cell. Or, for example, in what we call helper T cells, where a macrophage will take a protein that's outside of the cell rather than inside of the cell there, chop it up, present it to the T cell, which recognizes it or is instructed to recognize it. That then produces cytokines, which then act on another cell to go ahead and respond. So again, there are many, many different diverse ways in which the immune system functions in recognition. Now, what happens to a cell once it recognizes this bit of a protein that it's been instructed to learn to recognize? Well, it has on its surface, and this is sort of a cartoon a molecule we call a receptor. And if the part of a molecule that binds it, we call that a ligand, binds it, it then sends a signal into the cell. That's the membrane of the cell. That's outside, that's inside. And then we have a sort of Lego-like process where little bits clip on, one after the other, and ultimately form a little compound which then goes into the part of the gene just ahead of the part that will actually end up making the code for a protein. We call that the promoter or the enhancer. And once that binds on, that says it's a go to the gene. And the gene then gets turned on, and that may make another protein, let's say for the cytokine, that the cell would use to respond against the tissue. So there's, again, this very intricate process of recognition and response that occurs in the immune system. This is an example in cartoon form of that T cell receptor that we talked about, recognizing a peptide produced by, that's the MHC, that's the T cell receptor. And again, the first recognition is called LCK, or luck, meaning I found what I'm supposed to respond against. It then causes activation of another protein called ZAP70. That then binds to another one that starts the whole Lego-like process that ends on causing the reaction that occurs. And this is a cell over here, a T cell recognizing that dendritic cell. And what you can see if you just look over here are different molecules that we have been able to outline in color, showing you that the T cell receptor is in the center, other cells in the side over there. And so there's an enormous and interesting molecular process that occurs cell to cell, and it sort of is like a Ziploc bag where it pulls the two cells together to focus them when this reaction process occurs. Ultimately, many different pathways get activated, and all of them sort of, as you can see, start focusing down to that part of the gene that's going to get activated. And in this particular case over here, it produces what we call a cytokine, interleukin-2. And if all of these spots on that part of the pre-gene, so to speak, are filled in, that gene gets activated to then respond and start producing whatever it's supposed to produce. Likewise, there are pathways in the cell that can put little building blocks in here that block this process and prevent it from activating. And that's actually rather important in regulatory T cells that we'll talk about later that are part of MS. That, number one, it may block certain genes that we really want to turn on, and alternately, it may block genes that we actually want to turn off that will normally continue to produce whatever they're producing. So at a molecular level in MS, we're getting to the point where we're starting to begin to be able to map out pathways where the process that should be normal becomes abnormal. Finally, I've just put up, and this is very confusing, but just let me walk you through it. Let's say this is a receptor for a cytokine, and in the case that we're talking about, one called interleukin-10. Interleukin-10 is a good cytokine, and this is sort of a molecular model of how it's going to send a signal into the cell. And they just come in various flavors, which is all I want you to take home from this slide. So again, just to recapitulate, let's say this is a cytokine that then is going to activate a cell. It brings these two parts of a receptor together. It causes this Lego-like process that triggers the cell, that then goes on to the beginning of the gene and turns on a gene to produce those compounds that we talked about.